Hello and good evening, everyone. Welcome to night 2 of our Summer Research Experience student presentations. My name is David Dimitri, and I'm the program director here at Headwaters Science Institute. And in case you were not able to join us last night for night number 1 of our presentations, we've had over 20 students take part in our research program this summer. And tonight and tomorrow, they'll continue to share all of their hard work with you. So once again, tonight, you are also going to have the opportunity to meet their amazing mentors. Now, before we go ahead and jump into all of the action for tonight, I just wanted to let everyone know that the presentations tonight are a great example of what other students can do as a part of the upcoming fall research experience that we're going to have kicking off in September at Headwaters. So for family members, friends, teachers out there as well who are watching, as well as students too, this could be a great experience for a student you know or yourself if you're interested in joining us for our next research experience. So please be sure to check out all the details at our website, headwaterscienceinstitute.org, or at the link below. All right, so let's go ahead and we will get into tonight's presentations. So for those of you watching live on YouTube or Facebook Live, we welcome you and we encourage you to share any questions or comments that you have for our researchers as they're going through the presentations. And then after each presentation, our students and our mentors will have a chance to answer those questions from you. So now I'm very excited to welcome our first mentor, Andre. There he is. Hi, Andre, how are you?
Good. How are you, David? I'm
doing very well. I'm going to let you go ahead and introduce our first researcher for tonight. So take it away.
Awesome. Thank you. Yeah, thank you, everyone, for joining us. I just want to thank Headwaters for allowing me the opportunity to work with my mentee for the summer. And I got the fortune of working with Zach for the entire duration of the summer. That said, I really enjoyed being able to work with Zach. We had a pretty interesting project that we were trying to come up with earlier. And then, I think we came up with something pretty amazing, as an end product. Zach definitely is a, pretty gung-ho about trying to, do the work and, learning some new skills. And so it was definitely my pleasure to be able to, work alongside him and, try to guide him and really try to get, what we are produced here. But with that said, I won't take too much time away from Zach. And Zach, I'll just hand it over to you.
Great, thank you. Okay, my project is about understanding a potential cause of wildfire in California. And recently, California has started turning from a golden sunny paradise into a drought-plagued desert. Also, there's a new season in California from August to October, fire season. Some previous studies prior to the current drought in Northern California have found a relationship between weather in terms of precipitation and the number of large fires in the area burned. And because of the fact that there have been increasing drought and fires, I wanted to figure out if the current drought influences the severity and frequency of fires in Northern California. My hypothesis was that there is a correlation between drought severity and fire severity. In order to research this question, I needed to use some useful tools. I used QGIS to make all my maps and graphs that will appear shortly. I got my fire data through the California Department of Forestry and Fire Protection. And that data was extremely useful because it listed every recorded fire in California for the last 20 or so years. However, instead of looking at the fire data for 20 years, I looked at California's more recent drought from 2013 to 2020. I also used Google Sheets for some of my calculations. Since the drought has been getting more and more extreme over the last couple of years, and I love mapping, I mapped the drought by adding up weekly drought data from the US Drought Monitor. Every week, they give each county a score from 0 to 500, which corresponds from no drought to extreme drought. After adding up the scores for a county in a given year, I categorized them into different ranges, as you can see on screen. For some context, here's what a non-drought year looks like. I mapped, this year was El Nino in 2017, where some people thought that this prior drought was over. But after a few years, the drought just came back in full force. with 2021 projected to have an even higher overall drought severity than 2020. El Nino in 2017 was a short break in the current everlasting drought in California. This next series of maps shows the burn percentages of a given county in a given year. I made these maps by clipping each individual fire from the fire data for a given year, and I calculated its area. I then set a relative scale in regards to county size and turned it into a map. 2020 ended up being one of the worst years, with 7 counties burning 10 to 40%. And it's also important to recognize that during El Nino in 2017, where drought was scarce, many counties had a relatively high burn percentage. This final series of maps shows the relative fire frequency between counties. If a fire spread across multiple counties, I would count that fire in each county. I made these maps in a similar way as the burn percentage maps by clipping county fire data in QGIS. Here, I did a basic linear relationship between the drought severity shown in the first set of maps that I showed you and the burn percentages shown in the second map. Based on this graph, there is little correlation between the two variables. I then plotted the relationship between drought severity and fire severity, and again, the R-squared value was extremely low, meaning there's little correlation between the two variables. In my methodology, I did not find a relationship between drought severity and the fire data. The way I chose the data that I was going to plot, however, might not have been the best representation for a potential relationship between fire severity and drought severity. And also, future consideration could involve looking at other weather factors besides drought, like wind speed, yearly average temperatures, and humidity. Thank you.
Awesome. Thank you, Zach. Yeah, with that said, if anyone has any questions or comments, feel free to, to type them in the chat, and, we'll be happy to address that. In the meantime, Zach, I'll just kind of, open it up to you for a little bit. I would say if you want to go back to that final map image that you were planning to demonstrate. There you go. Yeah.
Yeah. Oh, I see what I did.
Awesome. I'm not seeing any questions or comments come in. Give it another couple seconds. Otherwise, yeah, I don't want to hold it up too much longer in case the other students definitely, need the time as well. But overall, I just want to reiterate, thank you again, everyone, for joining us. And thank you, Zach. I haven't said thank you to you yet. It's been an awesome experience working with you. And I really look forward to seeing, what the things that you're going to be doing.
Yeah, thank you. I had a really fun time working with you as well. I really enjoyed it.
All right. Thanks, Zach, so much for sharing that. For all of our folks and friends who are watching out in California, it's a very appropriate topic to be researching and talking about right now. So we're really glad to have you share a little bit of that knowledge with us. And as Andre mentioned, just great work all around this summer. And Andre as well, thank you so much for your mentoring with Zach. I know you two have worked really well together. So we appreciate your efforts as well. Thank you.
Thank you.
All right. So next tonight, I'm actually lucky enough to introduce our next presenter, Caden Lou. Now, I was not Caden's mentor this summer. Caden's mentor, Inga Kanti-Jerpi, unfortunately could not be here tonight to introduce Caden. However, I am very happy to introduce Caden. I'm going to pull Caden up here. And so Caden has actually been conducting his research while in Jakarta at the Jakarta Intercultural School. So with that, I will let Caden take it away.
Thank you, Mister. Wait.
Kaden, it looked like you
Wait, do
just
you know
had
how
it.
to?
Looks like you just had it sharing on the screen, and then you might have taken it away. So can you go ahead and pull up that screen sharing again? All right, it looks like it's coming up here.
Can you see it?
There we go. Perfect. All right, I'm going to let you take it away.
Okay. So for this experiment, my research question was what type of milk promotes faster seed germination? Milk is one of the essential resources of humankind. Every day, people drink milk to strengthen their bones or to eat it with their cereal before they go to school. But research by The Guardian found that 1 in 6 pints of milk produced globally is either lost or wasted. In total, about 128 million tons of milk are either lost or wasted each year. So is there a way to put those million of tons of milk to use? Yes, there is. And milk can actually be used for planting. Calcium, one of the main necessities for plant growth, can be found in milk and is used to provide structural support to cell walls. Thus, raw or unpasteurized, cow's milk has similar properties and benefits to both humans and plants. In my experiment, I want to find out if milk has a better germination effect on seeds than water. So for my research questions and hypothesis, the key questions I'm trying to figure out is, does milk provide the necessary nutrients for seeds to germinate? And what type of milk promotes seed germination? For my independent variables, they are air, which is the outdoor airflow, warm temperature, because in Jakarta, the temperature is warm, ranging from around 75 degrees Fahrenheit to 85 degrees Fahrenheit, depending on the day. And the type of milk used to germinate the seeds, which is almond milk, chocolate milk, soy milk, and regular milk. My dependent variable is how fast seed germination is for each treatment, and my hypothesis is that nutrient-based fluids will help seeds to germinate faster, and seeds watered with almond milk will germinate quicker than seeds watered with other types of milk. So for pre-setup considerations, the data I want to focus on is that my experiment will focus on finding out the falling stage, which is the third stage seedlings. And I use mung beans in my experiment because they are easy and quick to germinate. They are a popular food source in Indonesia. They're widely available and cheap. The liquid for my treatments are regular milk, almond milk, chocolate milk, soy milk, and water. So for materials and methods, my experiment was conducted in an open-air balcony with 5 different treatments and 10 replicates of each treatment. Each treatment was a plastic cup filled with 3 cotton buds and 10 similar mung bean seeds in it each. The 50 plastic cups were placed randomly in a 5x2 grid. After that, each treatment was watered with 10 milliliters of their respective liquids and 5 milliliters of water. However, the control treatment was watered with 50 milliliters of water. I will repeat this process 1 time every day to ensure that all of the mung bean seeds were getting enough of each of their liquids and not drying out. At the same time, I would also look out for whether the seeds have germinated or not, which was basically when the leaves were visible from the seeds. So these are the results. As you can see, 1 seed in treatment 8 of the almond milk treatments germinated, while the seeds in the soy milk, regular milk, and chocolate milk didn't germinate at all. However, as you can see from the water treatment, at least 5 seeds germinated. So in the end, this experiment both proved and disapproved of my hypothesis. It has shown that milk is not as good as a substitute of water when it comes to seed germination, as the water treatments were the only treatments that have germinated besides one of the seeds in treatment 8 of the almond milk treatments. However, almond milk is the best milk for seedling growth as it has germinated at least one of the seeds. This may have been the case because on day 3 of the experiment, the milk started to rot, and even in both the soy and chocolate treatments, the milk solidified and prevented seeds from receiving water. In conclusion, while almond milk is the best milk for seed germination, it shouldn't be used for seed germination. It shouldn't be used for seed growth. Other observations that may have impacted this experiment was that the amount of milk given to the seedlings may be too concentrated. because after drying, since the temperature is very hot during the day, the dried milk residue in all of the cups may have caused the seeds to hibernate rather than germinate. There was evidence of thick dry powder in the milk, chocolate milk, and soy milk cups. However, the almond milk did not seem to dry as quickly and leave heavy residues. Thus, the seeds could probably get water easily. So for future research, I would like to improve my experiment by expanding the number of liquids used for the experiment, such as vinegar, cane juice, and other types of nutrient-based liquids, to find out which liquid is best for seed germination. And I would also like to expand to other types of seeds and food crops, because they may react differently to the liquids used on them. And then these are my works I did.
All right, Kaden, thanks so much for sharing that research. Really interesting stuff that you had going on there with the plants. I will kick it off with a question. So you mentioned maybe trying some other liquids in the future. Do you have any hypotheses as to which of those maybe would have a better effect on plant growth than what you saw with the different types of milk? Maybe based on some of your background research that you did, or your knowledge of other types of liquids?
I don't think so, because I didn't do any research other than between the types of liquids I used.
Okay, good. Okay, another question we have actually is, do you think after the seed has germinated, would the fluids help plant growth?
I actually tried that as a side project, using germinated seeds. So the water, using the seeds that germinated already with almond milk, because almond milk was the best. Was the best based on the results, but it actually didn't work and ended up killing the plants.
Oh, interesting. Very interesting result. Let's see if we have other questions from the audience here. I guess one final question I would have for you is, what was the toughest thing about your project in terms of implementing it? Was there anything about the design or the setup that was kind of more challenging than other parts of the project?
I think it was having to repeat the experiment, because before I did this experiment with cotton buds, I actually used soil, and it turns out that if you mix soil with any type of milk, it would actually start to smell bad on the second day, and it will start to solidify more easily.
Gotcha. Excellent. All right, Kaden. Well, thank you so much for sharing your research, and thanks for all your hard work. I know with the time change and things from Jakarta, it has been excellent and awesome having you with us in your program this summer. So thanks so much. All right, we're going to bring up our next mentor. We're going to introduce Dan Dudek to bring in our next researcher. Dan, go ahead and take it away.
All right. Hey there, everyone. So if you didn't tune in yesterday, my name is Daniel Dudek. I'm a PhD student at the University of Texas at Arlington. I do a lot of work in genetics and ecology, in amphibians and reptiles. But today I have the pleasure of introducing Gia Key. She's coming all the way from New Jersey. So, you can participate in this program even from across the country, not just California. And she's doing important work, some really cool work on, how this sort of pandemic has been affecting all of us. So I'm really excited to, listen in and tune in. And I hope all of you enjoy her talk and presentation.
All right. So my research topic was on correlation between sleep quality and anxiety levels in 1 year of quarantine. And so first we have some psychology background, since there has been a misconception with the mind and body, with some people thinking that they are separate things and they work on their own. However, we found that our mental health and physical health are actually interconnected, and that biological factors actually impact us mentally, while psychological factors will also impact us physically. And so for those who don't know, anxiety is associated with feelings of worry and unease, and anxiety disorders are a class of disorders marked by excessive or chronic anxiety or apprehension. And to be clear, for my research, I didn't do anything into anxiety disorders, but more for the anxiety that we may feel in day-to-day life. And so more introduction into my research. General research, actually, without quarantine, has found that sleep deprivation can instigate or worsen anxiety disorders and vice versa, which means that anxiety can also make it difficult to sleep. And we found that according to sleep standards, that sleep habits actually changed a little bit due to quarantine. And they found that 68% feel stressed or find it hard to sleep even or after lockdown measures were lifted. And so why is this important? Well, we really do need sleep. A lack of REM sleep, or rapid eye movement, which is a stage of sleep, can actually cause increase for obesity, many memory problems, or make someone more prone to anxiety and depression. And so for my research question, I had the independent variable of sleep quality and the dependent variable of anxiety. And I hypothesized that there will be a positive correlation between sleep quality and anxiety levels. For materials and methods, I mostly use surveys. And so a Google form was distributed via social media for individuals to answer, and 32 individuals participated. The form included 2 surveys, which were the Pittsburgh Sleep Quality Index and the Self-Rating Anxiety Scale, in that order. Answers are translated into scores for each respective test, and a linear regression line was used to evaluate results in correlation. So more about the surveys, we first have the Pittsburgh Sleep Quality Index. And in this survey, participants were asked a range of questions that asked them to rate their experience with sleep, falling asleep, and their habits. Scores were given in components and added together at the end for a final score. And that final score ranged from 0 to 21, with 0 indicating no difficulty and 21 indicating severe difficulty in all areas of sleep. For example, component 1 was subjective sleep quality. So the question actually asked, how would you rate yourself on your sleep quality? And depending on the response, the participant would get a score ranging from 0 to 3. There are many components like this, and at the end we would add them together, and the score would range between 0 and 21. The next survey was the Self-Rating Anxiety Scale, and for this survey there were 20 questions, and participants were asked to rate themselves from little or none of the time, some of the time, a large part of the time, and most of the time. And so scores were given for each question, and depending on their answer, they would get a different point. So little or none of the time would be 1 point, some of the time would be 2 points, a little part of the time would be 3, and most of the time would be 4. After all of the questions are added together, then it should range from 0 to 80, with 0 indicating low levels of anxiety and 80 indicating severe levels of anxiety. And so here are my results. I found that there was an R square value of 0.02766 about. And so what that means is that if you multiply by 100 for a percentage, there would be 2.7 about percent of the variability in anxiety would be explained by sleep quality. And then I got an R value of 0.1663 about. And as you can see, it's a pretty small value. So it means that there isn't a strong correlation. But looking at the graph, it is a positive number and it is a positive correlation. And now we can look a little bit at the discussion. And as I mentioned before, the statistics suggest that even though because there is a low R value, sleep quality and anxiety are not strongly correlated, but we do see a positive correlation. And some things to consider are sampling and conditions. Due to it being summer, many students' sleeping habits may vary since there isn't like a schedule due to school or deadlines of any sort like that. The survey was anonymous, so to avoid bias due to fear of judgment for either sleep quality or anxiety, since those can be very personal sometimes. And then some other things to consider is that I compared my research with frontline medical staff. And in that research, they found that frontline medical staff exhibiting co-occurrence of anxiety and depression are associated with worse scores on sleep quality. And so comparing mine with that, for the most part, the general public were consistent with those results, but they weren't as significant. Finally, we have some future research. Because my study was only done about 1 year into the quarantine, depending on the Delta variant, or if we stay longer, a more long-term study could be made. Research could also be done with different countries, since different countries have different cases and different ways that they approach this pandemic. And so results could be compared to the number of cases per country for more correlational results, or just other variables in general. And lastly, a wider age group could be surveyed, because I did use social media to send my survey out. It mostly targeted towards people who obviously use social media, which are like teens or like more, I don't think there was any participant that was older than 50. So maybe elderly or younger children I missed might kind of sway the data, and that could also be researched. And that is all I have for my presentation.
All right, thank you so much, Gia. So what you're saying is essentially old people don't use social media at all?
Well, not necessarily. I just didn't really get to them.
Yeah. All right. So I just want to ask you real quick. So when you were coming across these different sleep quality indexes, what made you choose that Pittsburgh sleep quality index? Was there anything sort of intriguing about it, or?
I thought, because I mostly got inspiration from the frontline medical staff study that they made. I just used similar things so that it was more consistent and easier to compare their findings with mine.
All right, that sounds great. We'll give it 2 more seconds to see if any questions roll in. But so tell me one more thing. What was the most enjoyable part of this whole process, or doing your project?
I think because I've never really done research before, it was really interesting to get to experience something new and to kind of see what it actually is like. So yeah.
Oh, okay. So we have one more question coming in, but yeah, great. It was great working with you too. So I came into the presentation late. I can't come in late. So what percentage of the survey received a response?
I actually didn't really find the percentage of that, but I think maybe because I used my own social media, which is Instagram, so that has maybe 500 followers or so, and also David sent my form out to some other people that were participating in the research experience. So I'm not sure what the total number is, so I can't give you a percentage in calculation like that.
All right. Well, it's definitely a great answer. So when you don't know the answer, don't try and make up numbers. So that was a great answer. All right, but yeah, thank you so much for participating. It was great working with you.
Thank you.
Excellent work, and great words of advice from Dan there as a mentor. Excellent work, Gia, very much. That's really cool stuff. Very appropriate to be studying, I think, right now, and I think something we can all learn from. So thanks so much for sharing. And Dan, thank you very much for your help and guidance with Gia this summer for the program.
All right, thank you for having me.
All right. So yeah, a great example, again, as Dan mentioned, of what you can do if you are a student in New Jersey. We had Caden on last, who was joining us from Jakarta. So if you're interested at joining us for our future program with the Fall Research Experience at Headwater Science Institute, if you're a student watching, or you're a family member or a parent of a student who might be interested in doing some research like this, please feel free to check out more information at headwatersinstitute.org, and you can learn all about how to sign up for our program starting this September. All right, I'm very excited to bring on our next mentor who has joined us this summer here. Let's see if we can bring in Chasey. I'm not seeing her screen, her video. Let's try that again. So I think we're gonna pause momentarily and let Chasey leave the backstage area. She'll join us here hopefully momentarily to bring on our next researcher. All right, well, we may have lost our next mentor momentarily, but I will go ahead and actually then bring on our next student researcher, Rose Gallagher. Rose, welcome. So I know Rose has a really cool project she's going to tell us a bit about. She had some really cool experience at Weber Lake this summer in California conducting her research. And so what I'm going to do is go ahead and let her tell us a little bit more about what she studied and learned as part of that experience this summer. So Rose, go ahead and take it away.
So my project is how does human impact affect the water quality at Weber Lake? And again, my name is Rose Gallagher, and I worked with my mentor, Chasey Danger. So both pollution and water pollution levels are constantly increasing as a result of human impact. Water pollution statistics of 2021 state that over 2.5 million acres of ponds, reservoirs, and lakes are unsafe due to nutrient pollution. Different sources of pollution in our waterways can include fertilizers or detergents, power plant runoffs and dumps, or sewage runoffs and dumps. These sources of pollution can also affect our drinking water. As a result of human water pollution, the water quality of many lakes and other bodies of water is decreasing. Studying this is crucial because of all the long-term effects this could have on humans and especially healthy aquatic ecosystems. I conducted my project at Weber Lake. This land consists of approximately 3,000 acres and is located north of Truckee in between Perazo Meadows and the Jackson Meadow Reservoir. The Truckee Donner Land Trust acquired this land in October of 2012 from the previous longtime owners Cliff and Barbara Johnson, who had this land in their family since the 1870s. Up until this year, 2021, it had been completely closed to the public. The most serious water quality issues originate from human activities and land usage in or near a body of water. For this project, testing the quality of the water is important to see if humans are negatively affecting the waters. This helps the public be more conscious so they can prevent potential problems before they happen. Studies like this help reduce pollution and help keep aquatic ecosystems flourishing. My research question was, how does human impact affect water quality at Weber Lake? The independent variable was the human impacted locations and the non-human impacted locations. My dependent variables were the 8 elements of water quality: temperature, TDS, pH, nitrate, nitrite, and the total hardness, total chlorine, and the total alkalinity levels. I hypothesized that the locations with more human impact would have a lower quality of water. The materials I used to conduct my project were a thermometer, a TDS meter, a pH meter, and water quality test strips that tested nitrate, nitrite, total hardness, total chlorine, and total alkalinity. The 8 elements of water quality started with temperature. Water temperature exerts a major influence on activity and growth of a body of water and its ecosystem. It goes hand in hand with what kind of organisms can live in a certain body of water. A dramatic change in temp due to human pollution could be very harmful to the ecosystem. We also tested TDS. The total dissolved solids is tested to determine the general quality of water by measuring the amount of materials dissolved in the water. The materials could be metals, minerals, salts, and ions. In some bodies of water, the TDS is naturally low or high, so a dramatic increase over a short period of time would be an indicator of human pollution. We also tested pH. pH is a measure of how acidic a body of water is. A neutral pH is 7. The normal pH range for an aquatic freshwater ecosystem, which is what Weber Lake is, generally lies between 6 and 9 pH units. Pollution can cause a change in pH which can damage aquatic ecosystems. We also tested nitrate and nitrite. Nitrite are a salt or an ester and ion of nutritious acids which can come from fertilizers through runoff water, sewage, and mineral deposits. Nitrates are an essential plant nutrient, but in excessive amounts they can cause significant water quality problems. We also tested total hardness, which is the amount of dissolved calcium and magnesium in the water, as well as total alkalinity, which is a measurement of all the concentration of all alkaline substance dissolved in water, as well as total chlorine, which is measured because the levels depend on the turbidity of the water. All three of these elements could be influenced by human pollution. For my project, for 3 days, a group of 5 people and I split up and went out to 4 different areas of Weber Lake, 3 different times a day. Once in the morning, once around noon, and again in the afternoon. Each time we collected a measurement for all 8 of our elements being tested and wrote the measurements down. There were 2 human-impacted sites as well as 2 non-human impacted sites. The 2 human-impacted locations were the campground shoreline and the lodge shoreline. The 2 non-human impacted locations were the creek shoreline and the untouched shoreline. Once I had all my data logged into a spreadsheet, we averaged out each day's worth of numbers for each element and their location. Using the data table, we compared the human-impacted numbers to the non-human impacted numbers by using GraphPad to conduct a t-test to give us a p-value. This told us if the data collected is statistically significant. After, we gave each element a graph using Google Spreadsheets. These are the results of a graph. When you conduct a t-test, you get a p-value, and if a p-value is below 0.05, that means the data is statistically significant. Because these two were not under 0.05, they were not significant. Water temperature and TDS, again, were not significant, nor were the pH levels or nitrate, nor were nitrite or total chlorine levels. The 2 significant results were total hardness and the levels of alkalinity. The harder the water, the lower the toxicity of aquatic life. As we can see on the graph to the left, the non-human impacted total hardness is higher than human impacted. This tells us that there was less toxicity in the non-human impacted locations. For the levels of alkalinity, even though both numbers are fairly regular, this just shows us that the non-human impacted had more acid and acidity in the water. There were 4 sources of error in my data. First one was that the data was taken over a span of only 3 days and I had a limited amount of time to take my research. Another one was that there were 4 outliers in my data. This could have been because the group that helped assist me with my project had never worked or had experience with the tools we took measurements with, even though their help was very much appreciated. The 4th one is that the lake had only been open to the public for a year, which could cause less human impact on the lake. The future research I have for this project is because human impact is going to be more frequent with opening the lake, going back every year to do the same test would be a way to further the research. This would tell us how human impact is affecting lake water over time. The 3rd one is possibly collecting data in more than one body of water to compare and contrast. And the 3rd one is that human activity is proven to have a heavy impact on many bodies of water around the world. So continuing tests like these in other areas or continuing it over time would tell us more about what to do to solve the problem of human pollution in our waters and to keep our ecosystem happy and healthy. These are more excited, and thank you so much for listening.
Hi, Rose. Sorry, my Wi-Fi decided to cut out right when I went to introduce you, but you did a fantastic job. I guess my question for you, because I know you started this project looking to get some more experience in research. Has this been a good experience? Do you feel like you'd want to do this again next summer and pick a different project?
I think I would definitely like to do it next summer. It really opened me to more experience, especially being able to do my research hands-on instead of all online, which I was a little bit planning on doing. But turning against that was definitely a good idea just because I got so much experience and using all the different apps that I use and like learning how to take really intense like notes and stuff. It definitely was a very good experience.
I agree. That summer camp looked great.
Mm-hmm.
We can give people a second to ask some questions. Let's think. So if you possibly went back next summer to Weber Lake, would you consider using the same locations, or would it be something where you might want to use different locations?
I think it could be really important to use the same locations just to see if there was a difference and if there was more human impact over the year. But I also think I would test in other places and I would try and test turbidity because I think that's an important one that we didn't get to when we were at the lake.
Cool. All right. Well, this was a great summer project and I'm excited to see what you work on next.
Yes, thank you.
Mm-hmm.
Yes, Rose, thanks so much for sharing and for kind of giving us a little bit more insight as to what it was like this summer to conduct that research at Weber Lake. So thanks so much for your hard work and for presenting on that tonight.
Thank you.
You're welcome. All right, so we're going to keep Chasey on. And Chasey, you can go ahead and introduce our next researcher, but I'll also give you the floor to introduce yourself if you'd like beforehand, since we lost you there before bringing Rose on. So go right ahead. You can take it away.
Thank you. So my name's Chasey. I just moved into a new apartment, which is why my Wi-Fi decided to cut out right when I went to introduce myself last time. I'm a graduate student at UNR, and I was also Amy's advisor. And Amy had pretty much from the beginning a full idea of what she wanted to do. So it was just about helping her solidify an idea and figure out how we were going to research that idea. So I'll let Amy take it away and explain her summer project.
Okay, hello, my name is Amy, and my project is titled What Effect Does Human Population Have on Condor Population Over Time? And my mentor is Chasey Dancer. So condors are the largest bird in North America, and in the 1980s there were less than 2 dozen of them left in the wild. Their species is teetering on the edge of extinction. Everyone was super worried about them. But over 40 years later, they've made a full comeback with over 100 flying free just in California. But the question kind of still remains, is the species safe? And how are humans threatening them even now? So some ways that humans threaten condors, kind of the most prolific way, the biggest way is lead poisoning, which threatened them then, it threatens them now. And a lot of it is from lead-based ammunition, which is from hunting. So humans would shoot deer, the condors will eat the carcass, they'll eat the bullets, they'll get lead poisoning from that. That's a big problem. The other one is microtrash, where mother condors will actually feed their babies trash. The babies will be full of trash, and they can't really function well that way. And they'll also maybe get lead poisoning from the trash as well. And I'm not really sure why this happens. Probably something to do with the fact that other birds also feed their chicks like bones and stuff. Maybe they can't tell the difference. And so condor reintroduction programs were, that's basically how the species came back. Less than 24 in the wild, they captured them all, bred them in captivity, tried to release them once, but didn't go so well. Let them back, release them again, it went better. Now there are many flying free, and the species extinct. So more condor facts. Actually, the reason they're bald, the reason they don't have any feathers on their head is when they're head deep in a dead animal. They don't want all that bacteria and stuff to stick to their feathers and just make them sick all the time, and then just rinse their head when they're done and they're clean. It's a lot easier. Some other facts is they have a very slow reproduction rate, which was a big challenge in bringing the species back. They just reproduce so slowly. And another thing, they can store 3 pounds of meat in their crop, which is that big thing in their neck. And they actually pee on themselves when they're hot, and it cools them down. A lot of other vultures do that. So the 3 parks I chose to study for this, or just use for this, are Pinnacles, which is kind of more central California. It's 26,000 acres, and it's relatively new. It officially became a national park in 2013. And the Redwoods, which is more northern California, slightly bigger, 131,983 acres, also older, came apart in 1968. And then Sequoia, which is more southern California, the biggest, the oldest, came apart in 1890, a lot more acres than the other 2. And so my research question is, how have condor populations been affected by human populations throughout national parks in the last 10 years? And my hypothesis was, as human populations in national parks increase, condor populations will decrease. In other words, human populations will have a negative effect on condor populations. So materials and methods, it was pretty much, it was all online research. I really have to go anywhere for this. And so just online data collection, my computer, Google, and I use GraphPad Prism software to make my graphs. And then for methods, yeah, pretty much just that, searching online, analyzing the graphs. Just doing all that using the NPS visitor data search engine to find that data. So this first graph, just looking at human populations in the 3 parks over 10 years. And as you can see, the pinnacles is the lowest, redwoods next, then sequoia. So the park size kind of correlates with the number of visitors each year. And so this first graph, human populations and pinnacles over 10 years, R-square value of 40, so it kind of matches up. And then the condor population in California, so yes, just in California in general, over 10 years, R-square value is about 80, so it really matches up. So if you just compare these 2 graphs, and just you can see the lines, you can see the shape, which is, yes, this human population is going down, condor population is going up. And so these 2 graphs really support my hypothesis where human population is down, condor population is up. However, the other 2 graphs have a very low R-squared value, and they don't really match up at all. So that's redwoods, that's sequoia. So kind of in conclusion, I think my hypothesis was mostly disproved. The humans have a more positive effect, a more complicated effect on condors than I first theorized. The condor population data graph and the pinnacles data graph do support it. The other 2 just have such a low R-squared value that I have to conclude there is a lot more factors I need to bring into my research. And so yeah, so just for future research, it's probably just expanding on that, seeing what questions I should be asking to match my data, and just, yeah, further researching that hypothesis and just see what else is out there. And also lead poisoning. Like when I was looking into that, there was a lot of data for that. It's a whole other big issue, so it could also be really interesting just to investigate. And so we're excited. And thank you.
Hey, great job. Your presentation was so pretty. That was my favorite part about it, was just how well it was set up, all the pictures and stuff. So I think the biggest question, and I think you went over it a little bit in your research, was what we learned while we were trying to research condors, and how you would do it differently, because I know you're so passionate about ecology and conservation. So how have you been thinking about how we would have done that condor research a bit differently next time?
So yeah, that was the biggest thing, is the fact that we didn't have condor data specific to each park. We just kind of did generally California. And so, you know, trying to like message the rangers, they didn't really respond. So just finding a better way to do that, finding a way to get specific condor data and seeing how that might change the research.
Yeah, and you know, once you can get into bigger projects, it'll be great to bring in that lead poisoning element and the micro trash, and you know, you could do 10 years of projects on there. It looks like there is one question. It says, if we see a spike in park visitation after the pandemic hopefully ends, do you anticipate the condor population will still continue to rise?
Oh yeah, that's like thing came to the conclusion where if I was just looking at my hypothesis, I'd be like, oh no, more people, less condors. But now that I've seen the other 2 parks and I've thought about the condor reintroduction programs, and maybe all these visitors are bringing in money that can help the condors, maybe they will still continue to rise. So I don't know.
Maybe humans aren't that bad, huh?
Yeah, maybe they will rise, and maybe humans do help condors more than I thought they did.
Yeah, and I mean, the condor reintroduction programs are new, so it might be something that they're so new and people are so passionate about them that humans almost have a very beneficial effect on condors because they're such a big ticket. You know, you love the way they look, you think they're cute and stuff, and I'm sure other people do too. All right, awesome job. I loved that presentation.
I agree. Great job, Amy. That was awesome. we had a comment from one of our fellow mentors saying awesome pictures too. So always great to see some beautiful pictures of condors in there too. But great research too, very appropriate as well right now for everything going on. And it'll be really interesting to see what happens with the population over time now as we hopefully get out of the pandemic, and we'll see what the park visitation numbers look like too. Thanks so much for sharing. And Chasey, thank you so much for your help and guidance with Amy all summer long. We really appreciate your work too, with Amy and with Rose too, from our previous speaker. Thank you. All right, so I'm gonna bring on our next mentor, Tara Webster. She's gonna join us here to introduce our next student researcher from the summer. Tara, take it away. Oh, I'm sorry to interrupt you. We don't want to miss out on anything. You were muted. Yeah, go right ahead.
I was muted. Thank you. My name is Tara Webster. I was the mentor for Nithya this summer, and I am very excited to see her presentation. So I'm going to actually let her introduce what her work was about, and I will help her with her questions after.
So like Tara said, my name is Nithya, and today I'll be talking about how recycled gray water affects plant growth. So clean water is becoming scarce, and there has been an increase in droughts due to climate change. To the right, you can see a graph of the average temperatures in July, and you can see that the temperatures have been rising due to climate change. So due to water shortages, there have been limitations for residents to lower their water usage. And so an alternative for water would be gray water. Gray water is excess water from toilets, sinks, washing machines, and all your appliances at home. Which leads to my research question. How does recycled dishwater, aka gray water, impact the growth of outdoor perennial and annual garden plants? So on the left, you can see how I laid out my plants. I tested 2 species, petunias and marigolds. I did this in my backyard in Northern California. I tested out 3 water test groups: tap water with dish soap, and gray water for my family's dishes. Every day, I'd give 2 tablespoons of water in the morning and evening to each plant with their respective water type. In the evenings, I'd measure the plant growth by placing a piece of cardboard on the pot, and I'd put the ruler on the piece of cardboard and measure the highest point, as you can see on the right. So my hypothesis was that there would be no difference between the group medians. And to figure out if this was right, I used an ANOVA test, which would basically compare 3 or more numerical test groups. So as you can see, we figured out that petunias between the 3 test groups, that there was no difference in height, as our p-value was less than 0.05. And you can see the same result from the marigold group too, as both of them were also less than 0.05. From our research, we found out that soap water and dishwater don't have a negative impact on plant growth. This then suggests that recycling dishwater might be a good alternative for watering household and garden plants. Some potential sources of error could be that the plants were relocated during the experiment, and there could have been inconsistencies in measurement. The concentration of soap and other dishwater contaminants could have changed, for example, cleaning chemicals in the dishwater. For further research, I would like to figure out a way to conserve water as a city-based feasible solution, and some other gray water alternatives such as rainwater from rain barrels or bath and sink water. And I'd like to thank Headwater Science Institute, my mentor Tara, and my parents.
Wonderful. Thank you so much, Nithya. It's really great working with you this summer, and I'm very proud of the work that you did. Are there any questions? So one of our questions is, what was the toughest aspect of this research that you faced?
I think when I was measuring each, like I had 12 plants, so I'd have to spend around half an hour to 45 minutes every day measuring it. And I felt like sometimes either I wasn't getting the right measurement or, like I said, inconsistencies of measurement. So I felt like sometimes I'd get like a centimeter or 0.1 of centimeter plus or minus 1 off. And so I felt like that could have been another aspect of that made my results a bit different.
Wonderful. Any other questions?
All right, Nithya, thank you so much for sharing that research. I guess one question I would have for you is, so if you were to be able to continue this research kind of on to the next step, say you could do another 10-week program, what would be the next kind of most important thing that you think would be worth addressing and researching to help further your research as you've presented tonight?
I think broadening the experiment would be helpful with maybe more species and more water types, just because this was such a small, like for less than 4 weeks. So I couldn't exactly get something that I could use for a community or, other species. So maybe hopefully doing more plant species if I were to do this next time.
Excellent. Great. All right. Well, thank you very much for sharing your research. Tara, thank you so much too for your support with Nithya all summer long and for your guidance on her research. We really appreciate your help out there. Thank you.
Wonderful. Thank you.
All right. And last but certainly not least, I'm going to bring on our final mentor of the night, Heather Raymer. Hi, Heather. Heather is going to go ahead and introduce our final researcher for tonight's night of presentations. So Heather, go ahead, take it away.
Hello, everybody. My name's Heather Raymer. I use she, her, hers pronouns. I'm a PhD candidate at the University of Virginia. And this summer, I have spent my time mentoring Nicole on her project. Nicole is going into the 10th grade this year and lives in the Bay Area. And we've had really good time pursuing her interest in viruses after she got introduced to them in her biology classes this last year. So with that, I'll let Nicole give you the rest of the talk.
Hi, yeah, my name is Nicole, and I researched viruses, particularly the spread of viruses. So for an introduction, a virus is a protein filled with its genetic information in the form of DNA or RNA.
And Nicole, I'm really sorry to cut you off there, but we don't have your specific slides on the screen. Can you go ahead and try sharing that again along the bottom?
Yeah.
Just want to make sure we have the audience able to see all of your research there. All right, I think we have it now. Let me go ahead and bring that up. There you go. All right, let's go ahead and we can start things off.
Okay, thank you. Like I said, I decided to research viruses, particularly the spread and predictions of the spread. So for an introduction, viruses are proteins that are filled with their genetic information, which comes in the form of DNA or RNA. They're similar to cells, but they can't replicate alone, and therefore they infect other cells to do so. I also researched the R0 for these viruses, which the R0 value is how contagious a virus is. To put it simply, R0 value is the number of people that an infected person gives the virus to. So if the R0 is less than 1, the virus will die out on its own because it's been given to less than 1 people on average. If it's equal to 1, it will stay the same, and if it's greater than 1, that means it will get worse over time. By comparison, the R naught value to the viral properties that I chose, we can see if any of the viral properties affect the R naught value and see if it maybe can be predicted. The viruses that I chose are SARS-CoV-2, the variola virus, influenza virus, Ebola virus, morbillivirus, hepatitis B virus, and the polio virus. My research question was, are R naught values correlated to the following viral properties? My independent variables were capsid structure, genetic material type, size, shape, enveloped, and segmented. These are the different properties that I chose. And the dependent variable is the R naught value. I hypothesized that there will be a correlation between the viral properties and the R naught values. For my materials and methods, I used pre-existing data sets since this wasn't really something I had the tools to research off of a computer, so there were no field materials used. I found the viral properties for the methods. I found the viral properties and the R naught values, and I analyzed them. For the data analysis, there weren't enough variation in the viruses for 3 of the properties, for enveloped, segmented, and the shape, to having meaningful analysis. It just means that there wasn't enough, like for example, for enveloped, all of the viruses were enveloped except for polio, so to compare the two wouldn't have we wouldn't have been able to find a pattern there. The other 3, the genetic material type, the capsid type, and the size, were turned into graphs, and those were able to be analyzed through that. For the graphs, I used t-test, ANOVA, and linear regression to analyze the data so I could turn them into graphs. For the results, the first one is the mean R naught value by capsid type with standard deviation bars. So this is comparing the capsid types. There are 3. There's helical, icosahedral, and complex. The graph shows, again, the means for the different capsid types, and the p-value that was found was 0.8761. And since this is larger than 0.05, this means that the data was not significant, meaning that the capsid type of the virus does not affect the R naught value. The next one was the R naught value by the size of the virus. And it was calculated in volume since there are a couple of different shapes. The p-value was 0.5432, and since it was, again, bigger than 0.05, it was not significant, meaning that the size of the virus does not affect its R naught value. The third graph was the genetic material type, so DNA or RNA, and the p-value is 0.9743, and a t-test was used, but this again was not significant. So the genetic material type as well does not affect the R naught value. So for the discussion, so none of the graphs has statistically significant patterns. So none of the properties affect the R naught value of viruses. My data did not end up supporting my hypothesis since I had originally hypothesized that there would be a correlation, and there was not. So something that might have messed up some of my data was that there might have not been enough variation of the viruses. Most of the viruses that I chose were pretty popular. You probably know quite a few of them. And since they're more popular, this means that they had a greater damage, which means that they had larger R naught values. So there might have been some of these properties that were properties of, like if a lot of them had the same property, it could be because that property did correlate to a larger R naught value, but there might not have been enough variation for us to actually see that in the graphs. And there might not have been enough viruses either. As you saw before, I chose 9 viruses, so there might have been patterns. I might just not have had like a wide enough range to see them all. For kind of the point of this, or something that it can be useful for, is that if properties that do affect the R naught value are found, then the R naught values may also be able to be predicted. The viruses, R0 values may be able to be predicted, which would be very helpful in seeing how contagious a virus is going to be able to predict what's going to come. I was able to rule out 6 of these properties, the 6 that I did, so the other properties can be looked at. For future research, more properties can be looked at, kind of similar to what I said before, to see if they affect the R0 values of viruses. And the same properties could also be looked at again with more viruses and a wider variety of viruses to see if anything was missed. So finally, for my works cited, I did everything online, so there were quite a few of them, but that is my presentation, so thank you.
So I just want to start off saying, Nicole, really great job. Made a lot of work on that even since we last met. And I've had the utmost joy and really enjoyed working with you this whole summer. So thank you for putting in all the work. And I guess at this point, we'll see if there's any questions.
Research I hope to study in the future. I know that I want to do science in the future. I know it's really broad, but I really do like kind of more human oriented, not really doctor per se, but I did really enjoy this experience, and viruses are something I'm really interested in. So hopefully something to do with that.
I see at the bottom someone's guessing you might be a future virologist or epidemiologist.
Maybe. That does seem really, really interesting. And I mean, hopefully that seems like it'd be really cool.
Awesome, Nicole. We hope to see maybe you will go down that path in the future based on some of this research. That'll be really exciting. Nicole, thank you so much for sharing that research. Really interesting stuff. And as Heather mentioned there, I think you've put a lot of hard work and effort into this, and especially over these last few weeks. So great job with your presentation and your research all summer long. And thanks so much for sharing it tonight. And Heather, thank you so much for your help and guidance as you've mentored Nicole through this summer and through this project. We really appreciate your dedication as well to all of the research that she's been taking part in. So thank you.
Thank you.
All right. So we're going to go ahead and wrap up another fantastic night of our student presentations. So I just wanted to thank all of our student researchers tonight again for their hard work throughout the entire summer. We're very proud of each of you, as everybody who has been watching tonight can attest to. You've each put in a ton of hard work throughout these past 9 weeks of the summer. And so we're just really proud of you for everything you've done throughout the entire summer. Now, for those of you that are watching that still have a question or a comment for any of our researchers, please be sure to drop those on YouTube or Facebook, wherever you're watching, and we'll make sure that our students get a chance to see those. One thing I did want to leave us with, again, if you're interested as a student or you know a student who may be interested in joining us this September for our fall research experience, please be sure to head over to headwaterscienceinstitute.org to find more details. Again, this is a chance for you to join in with some of the same research and having the same opportunity and experience to present on that research as part of our fall research experience. So we encourage any student who is interested in conducting their own student-driven independent research this fall to please take a look at headwaterscienceinstitute.org, and you can find all the details there for our fall research program. All right. And so in addition to obviously a big thank you to all of our students for their hard work this summer, I also wanted to thank each of our mentors for their continued guidance for our students all summer long. And then finally, thanks to all of you out there for watching. Just a reminder, tomorrow we're going to return again at 5:30 p.m. Pacific for the final night of our student presentations. So please be sure to join us then. All right. Good night, everyone.